Gene therapy is the introduction of genetic material into the cells of a patient to treat or prevent human disease. Gene therapy has been hypothesized for many years, but especially during the last 10 to 15 years, we have seen significant advances in viral vector technologies that make us now witness significant patient benefit. Compared to more traditional modalities, gene therapy has the potential to provide long-term benefit to patients, change disease phenotypes, even cure some patients, and that often in devastating diseases that have no treatments currently available to them. One very common vector used in gene therapy is called AAV, which stands for adeno-associated virus. It's a virus that is found in mammals and humans, but especially in humans is not associated with any known pathology. And it has been shown that it's very powerful in transducing even cells which are not currently going through the cell cycle and replicating, and as such really enables the treatment of humans in vivo.
The initial wave of gene therapy is seeking to address monogenic disorders. So a monogenic disorder is a disorder where one gene has the defect and 100% of the patients with that gene defect get the disease. That is a monogenic disorder. It makes it simple from a gene therapy standpoint because you know exactly what gene you've got to replace, and you know by replacing that gene you should be able to address the disease.
Initially, gene therapy is being used in monogenic disorders. These are diseases where typically one gene is faulty in the body of a patient, and this leads actually to usually very severe diseases that have typically few treatments available to them, for surely fewer treatments that you would call disease modifying. Obviously, by gene therapy, by introducing a healthy gene copy into the cells of those patients, has the ability to correct very significantly the disease phenotype in these patients. And in some cases, this can be as extreme as completely functionally curing the disease phenotype in these patients over the long term with a single treatment.
I think very simply, back in 2012, 2013, when we saw the initial gene therapy data sets, it was profound. It was a step change in how these patients were treated. In a lot of diseases, there were no treatments, so more than a step change. And so it wasn't the fact that it was gene therapy as a technology, it was the fact of what it could do to a patient. And as we started looking, it became apparent that there were a huge number of diseases that this would be applicable for. And so we wanted to put a strategy in place that would address it all. I'm excited about finding the next gyroscope. So what I think gene therapy has done in the last 5 years is look at these single gene disorders, and it's made real headway there and established itself as a modality that will deliver real products that will be marketed, that will benefit patients. I think its promise is much greater than that. I think the course to find those particular indications that are sufficiently validated and well worked out that they justify a gene therapy investment.
Gene therapy at the moment is focused on high-risk patients and patients that are sort of beyond the treatment of conventional therapies. I think also going forward we will see that conventional therapies and gene therapies will more and more overlap as gene therapy will become a staple of the 21st century medicine.